this is Unit 2, Population and Community Dynamics. And you've probably heard the word population and community several times before in your life. What we're really doing here is taking the idea of genetics, but expanding it. We're not just going to talk about one individual or one small family. We're going to talk about larger scale, larger groups, and how the frequency of an allele in the entire gene pool might be different than what it is in one small little family. So the first 3 lessons collectively will be about Hardy-Weinberg. They are 2 fellows who together came up with sort of a theory or a principle about how genes and alleles work in populations. We'll start by learning about just basically what their principle is, what the equation that goes with it is. We will do some practice, and then we will do a little lab activity to help us understand further how the Hardy-Weinberg principle works. It will, but it's not all the sections of a lab report write-up. It's more or less observations and analysis that we'll be doing. So the first thing that we have to start off with is just a brief definition of what each of those 5 terms means. Now, a population is something that I know that you would have defined in other biology and science classes. Does anyone want to volunteer, or does anyone remember what the definition of a population is? Organisms in the same place is what she's getting at. That would be certainly something that's important about a population. There's something else that we would want to add to this, yeah? No, she's right. At the same time, because, right, I could be at the same place as another organism, but 1,000 years later. And there's one more thing that has to be the same. Same species. So a population is defined by those 3 things. Same place, same time, same species. Now this is important because if you are not the same species as another organism, the chances of you having the same genetic code are pretty low. So we're going to be talking about populations, but we are also going to talk later about communities, which goes out further than a population. The second thing is the gene pool. And I know I always think of a swimming pool when I think about this, even though that is not what we are referring to. The gene pool, if we wanted to define it, is the sum of all genes and alleles in a population. So basically we're saying in a group of organisms who live in the same place at the same time and are of the same species, what are all the possible genes and alleles that there are? Then we have 2 words that you already know, phenotype and genotype, but we've added the descriptor frequency. So does somebody want to tell me what frequency means? How often it occurs. So phenotype frequency is how often a trait shows up. That one is usually easy. You look at an organism. Do they have that trait? Yeah or no? Genotype frequency is a little bit trickier. Genotype frequency is talking about how often a genotype, and by genotype we mean combination of alleles, shows up. Genotype frequencies are harder, as you saw in your family tree, because sometimes you don't know what someone's genotype is just by looking at them. You might know part of their genotype, but not the whole thing. The last one is allele frequency. So frequency continues to mean the same thing. How often? And here we're just talking about an allele is present. And I'm underlining the is present because we know you can have an allele but not show the trait. And so it's really difficult to figure out allele frequency just by looking at organisms, especially for the recessive allele, because lots of organisms who have the allele won't actually show it. So I'd like to ask you, if you have your textbook, I'm going to put what is essentially figure 19.3 on page 680 of your textbook up on the screen. So this is from your textbook. And I just want to show you how we might go about figuring out some of these numbers. So again, this comes from page 680 of your textbook, and it's to try to illustrate how we might figure out some of these frequencies, and so that you could see some principles about frequencies that have to be true. In this example, I have some organisms of different phenotypes. You can see there are two phenotypes at the beginning of ants here. They look the same, right? They have the dominant allele. But there's one at the end who has two copies of a recessive allele and has the recessive phenotype. Now, just by looking at these two organisms, I would not know whether they were homozygous dominant or whether they were heterozygous. I would need to know about their offspring to figure that out. So let's say I figured it out. I counted all the baby ants and I figured it out. And I counted how many there were. That's the number that you have, the population size. And the letter N will often represent population size. We'll see as we go through this unit, there will be some calculations here, some calculations there. And it's important to know what the total size of the population is. So if I take a look and I add up how many organisms there are 200. That is the total size of the population. 72 are homozygous dominant, 96 are heterozygous, and 32 are homozygous recessive. So if I was going to figure out the frequency of a particular genotype, I would take the number that have that genotype divided by the total population that exists. So this is mathematically how you would figure this out. It is really common for us to write frequencies as a decimal number. So 72 out of 200, or if you do quick math, out of 100, it would be 36 out of 100, is 0.36. So we could represent frequencies as percentages in this case. Unlike with genetics in the last chapter, it's uncommon to use fractions here. It is way more typical to use decimal numbers. We'll do the same thing for the next one, 96 out of 200. If we do that, we're doing 96 divided by 200 or 48 divided by 100. So it gives us 0.48. Here we have 32 out of 200 or 16 out of 100, 0.16. When we consider frequencies, I want you to notice what would happen if I added them together. They have to add up to 1. We're saying how many out of the total have this trait. If you ever calculate frequencies and the total is more than 1, something has gone amiss. It has to add up to 1 because we're always comparing to the total number. There's no way that you should have genotypes that add up to more than 100% of your population. Now, if we move to the next step and we ask ourselves, okay, how many alleles does that actually represent? Someone who is homozygous dominant for a trait has 2 copies of the dominant allele. So in this one, we're going to say there are 72 organisms, and each of them have 2 copies of the dominant allele. So that gives me, over here, 144. of the dominant allele. The organisms who are heterozygous have one of each allele. That tells me that I have 96 copies of the dominant allele and 96 copies of the recessive allele. So if I was trying to figure out the frequency of the dominant allele, I have to consider organisms who are homozygous dominant, they each have 2, and organisms who are heterozygous, they each have 1. When I add that all up, I get 240 big N's. Now if I continue to consider my recessive alleles over here, I have 32 organisms that each have 2 recessive alleles. So altogether, that makes 64 of the recessive allele. And if I add my 96 and my 64, I get 160 of the recessive allele. Now, so far, that's just told me how many I have. So if I want to know allele frequency, I ask myself, what's the total? How many alleles are there in this gene pool? Since you have 2 of each gene, whatever the number of the population is, so in this case it was 200, the number of alleles is double that, because everybody has 2 for each trait. So there are 400 alleles in this gene pool. So to figure out the frequency, I divide these both by 400. The frequency of the dominant allele is 0.6. The frequency of the recessive allele is 0.4. Notice how the frequency of the alleles doesn't match up perfectly with the frequency of the genotypes. But notice how if I add these two together, it adds up to 1. Frequencies have to add up to 1. All right. Now, the probability of you having to do this exact thing is pretty low, but I find that if I don't show you where we get these numbers from, and I just start throwing around allele frequency, you're like, what are you even talking about? Okay? So, now we'll talk about the Hardy-Weinberg principle. The Hardy-Weinberg principle is their explanation, so it's 2 guys, Hardy and Weinberg, to explain how recessive alleles like blood type O, do not disappear. A lot of people have the impression that if an allele is recessive, it's just going to disappear one day. People will just stop getting it. But one of the things that maybe you noticed, if you asked anyone in your family, is that blood type O is really, really common. Even though you have to have 2 copies of the recessive allele to have blood type O, it shows up a lot in the human population. So, the Hardy-Weinberg principle, which I'll show you is really based on a set of equations, is based on some conditions being true. So that you know these conditions are tough to meet, and we will talk about, and that's kind of what our lab will be about, how tough it is to meet these conditions. The first one is that it has to be based on a large population. We saw in the example with a pedigree about blood type that when you don't have a lot of data, sometimes things don't happen the way that you would expect them to. The reason the population needs to be large is so that random events don't have a large effect on frequencies. So what I mean by that is, smartboard. Here we go. What I mean by that is, let's say we have a population of 10. If something happens to 1 out of those 10, that's 10% of the population already. But if I have 100,000 and something happens to 1 of them, the effect on the overall gene pool is going to be really minimal. The second thing is that Mating is random. And I'm just underlining random here. That means it's totally by chance. There is no selection at all, no reason that one mate chooses another mate. Humans clearly do not follow this unless you are blindly walking in a cave and you mate with the first person you run into. Even then, you all had to be blindly walking in a cave. So, tough to meet this thing. The third thing is that there is no net mutations. And so I'll just specify what I mean by the word net. I'm not saying that there can't be any mutations, but when we consider the entire population, maybe some will be mutated one way, some will be mutated another way, and it'll kind of cancel each other out. So no net mutations means like overall. Sure, there can be mutations, but nothing is really changing in terms of which alleles people have. Lots of people could be having silent or point mutations. That often doesn't have any effect at all on their genetic code's translation process. Number 4, there is no migration. And by migration, we mean immigration or emigration. And it's starting to feel like social studies in here all of a sudden. So immigration means no new members enter the population. Emigration means no members who were in the population leave. And then the last one is that no natural selection against a phenotype occurs. So what that means is nothing happens in the environment that would cause a disadvantage to a particular trait. So you probably look at this and are like, this is never going to happen in real life. I concur. These are the conditions for which their formula is valid. And so even though we know that these conditions are hard to meet, we could still use their principle as a guide, not as a hard, concrete rule like this is how it's going to be, but to give us an idea of what's going on in a population. So what I'm saying is this, and I'll just say this again. We are not going to use their principle as like a, this is how it is. This is how things are. What we will do, though, is assume that for calculation purposes, so when I want you to calculate something, the question will say, we're going to assume the conditions of the Hardy-Weinberg principle are met. But lots of questions will ask you, hey, why did this not work out the way that we thought? And it's because one of these ridiculously hard to meet conditions wasn't met in a particular population. Okay? So here are the two formulas that you will need. The first one, which looks crazy and ridiculous, the first one is the Hardy-Weinberg equation. And you might be like, oh God, that is so long. Why do I have to remember this? And I would say to you, hey guys, look at your data sheet. It's on there, so you don't have to memorize this formula. But all that's on your data sheet is the formula. It doesn't tell you what any of the things mean. So what you have to know is what all of the things in the equation actually mean. So in the Hardy-Weinberg equation, there are P's and Q's that end up representing dominant or recessive alleles. So if you look at P squared, doesn't that make you think, hey, there's two P's there somewhere? P squared represents the homozygous dominant frequency. The p squared is what we use to represent homozygous dominant, people who have 2 copies of the dominant allele. The middle part, 2pq, so there's a p and a q. So the p is representing dominant, the q is representing recessive. This represents heterozygous genotype frequency. And then the q squared at the end will represent the homozygous recessive frequency. So that equation is all about genotypes. So we're looking for, in a particular population, what is the frequency, not how many people have it, but what's the frequency. So to figure out frequency, you would certainly make sure that you knew the population size and how many in the population had it. The total has to be 1. In other words, 100%. You can't have more than 100% because otherwise you didn't consider your entire population. Now the second thing is not on your data sheet. The second thing is the principle that I showed you earlier. This shows you that the frequency of the dominant allele, which is P, and the frequency of the recessive allele, which is Q, have to add up to 1. So sometimes a question might only tell you the frequency of the dominant allele, but you could figure out the frequency of the recessive allele because you know they have to add up to 1. Now, one of the things, and this kind of goes back to what Michael asked me earlier, are these like conditions that you can just ignore, or do they have to be met? For this equation to technically be valid, all of those conditions have to be met. If that is true, and this equation can be used, we will often say that we are in genetic equilibrium. In genetic equilibrium, P and Q are stable, so they don't change. Generation to generation, they are the same frequency. So if we were to calculate, like I did with the ants, that the frequency of P was 60% or 0.6, and the frequency of Q was 0.4, it would stay that way in every generation. Microevolution happens when P and Q change over generations. Notice how I didn't say evolution, I said microevolution, which means small changes in a population.